This is Agriculture Today, and I'm Shelby Varner with the K-State Radio Network. Beginning today's show is K-State's Kelsey, Andersen, Onofre, Sarah Lancaster, and Romulo Lollato as they discuss concerns with volunteer wheat. Kelsey continues the show and conversation about volunteer wheat as she reviews resources for producers. We are then joined by Brian Lubbers from K-State's College of Veterinary Medicine with information about the importance of a plan when it comes to handling dead livestock. K-State horticultural entomologist Raymond Cloyd finishes today's show as he discusses several pests that are currently active in the home landscape and garden, including squash bugs, blister beetles, cucumber beetles, and squash vine borer. That and more is coming up ahead on Agriculture Today. You're tuned in to Agriculture Today, and we start our show discussing volunteer wheat. And to join us on that topic, we have K-State Weed Management Specialist Sarah Lancaster, Wheat Pathologist at K-State Kelsey, Andersen, Onofre, and K-State Wheat Production Specialist Romulo Lollato. Thank you for taking the time to join us today.
Thanks for having us.
Thanks, Shelby.
Thank you.
Volunteer wheat is a concern. And Kelsey, could you start with us sharing of why it is a concern this year?
Yeah, so there's probably a lot of reasons why we're concerned this year about volunteer wheat. Maybe I'll just talk a little bit about wheat streak mosaic. One of the big reasons why we're really watching volunteer right now. If you're in Western Kansas or even Central Kansas, you're probably no stranger to wheat streak mosaic viruses. They can be pretty severe. And as you know, if you've experienced loss from these viruses, there's really not much you can do after you've had some infection in your field. So as a reminder, we're actually thinking not just about one virus, but a suite of viruses. We have wheat streak mosaic virus, we have triticum mosaic virus, a virus that we call wheat mosaic virus or sometimes high plains disease, and a few others like brome mosaic virus, for example. And they're all vectored by that very tiny wheat curl mite that's almost impossible to see with your eye. But that curl mite survives and feeds on living tissue. It stays alive and reproduces well when we have warm conditions at the end of the season. And when our plants dry down and we harvest, that tiny curl mite is looking for somewhere to go, right? So it's usually looking for ditches and some grasses, but it really likes to survive on volunteer wheat. So if we have wheat that we lost during harvest or due to hail, that wheat can emerge and serve as kind of a bridge until the crop is planted in the fall. And then that curl mite will move to that emerged crop and can infect that emerged crop with wheat streak mosaic. So that's why we're really concerned about the green bridge, right? That period where we have that volunteer out in the field, and we're really concerned about managing that this time of the year.
And Romulo, were there some factors from last year that are impacting this year's volunteer wheat?
Yes, absolutely, Shelby. I always like to think back on what happened last year so we can try to contextualize the problems that we're having today, right? And a few of the things that we can think of that were more statewide, one of them was we had a fairly cool and moist grain filling period. So the crop was quite stressed until mid-May, and then temperatures turned down, and we had some rain as well. That really created a lush of late-formed tillers. So we had very differing maturities between the primary tiller and the secondary, tertiary, or orth tillers coming out of a single plant. And many times the first ones, they were getting to harvest maturity, whereas the late ones, they were still a few growth stages behind. Now, what happens there is that the germ in the wheat seed, it forms fairly early in the seed development process. And once the germ is formed, it can emerge. And so that difference in maturity between the early ones that were stressed early on and the late ones that had a more cool and long filling period created a lot of harvest losses, right? Actually, it's very hard to calibrate a combine for that. Either we're waiting too long and we're losing, shattering, or we need to make adjustments to blow in the back of the combine some of those higher moisture grains as well. So that was one of the reasons why we're seeing quite a bit of volunteer. Another reason was just hailstorms. Starting in that mid-May time frame, there was a couple of hailstorms that came by in the central to southwest part of the state. And then later there in June or so, we had a few different hailstorms coming by the northwest and north central part of the state. Now again, the wheat was well into the reproductive stages. That germ was already formed in those grains as well. And even though we're very careful whenever we're planting a wheat seed to make sure we have high quality to improve our chances of a good stand establishment, truth is that once that germ is out, it has a very likely chance to germinate. And so all those hailstorms that we had across the state was another factor increasing the frequency of fields with volunteer wheat now. Again, because all of that grain kind of got lost in the field before it could be harvested. So I would mention those two major factors that actually led to just grain losses in the field.
Kelsey, you mentioned the green bridge, and Sarah, something that we might not want to keep going is the green bridge. And so what are some ways to possibly destroy it?
There's lots of things going on in a wheat field after harvest, right? Lots of things that farmers are going to be thinking about trying to keep out of the wheat field. So volunteer wheat is just one of many things that we're trying to spray. Fortunately, volunteer wheat is pretty straightforward to control. Glyphosate does a good job of controlling volunteer wheat and a lot of other grasses, as does clethodim and those grass-killing products from herbicide group 1. We also know that atrazine can be useful for some residual activity there for folks going to corn or sorghum next in their rotation. Other things to think about is utilizing this application where you're targeting volunteer wheat to go ahead and lay down some residual herbicides that might help with Palmer amaranth or kochia or other products. And so something like a flumioxazin or a Valor type of product could go a long way there as well.
Sarah, so it's worth thinking ahead and maybe trying to get a little bit ahead of the ball game if you're spraying down volunteer wheat.
That's right. Anytime you can get a 2-for-1 deal, it's a good day.
Sarah, for growers who are planning on going out and spraying, is there a certain time of when you think this is when most of my volunteer wheat is going to pop up, so it's the most effective?
So, it's one of those, it depends answers in terms of rainfall and things of that nature. As we think about the green bridge, it's not maybe so much the importance of when the biggest flush comes up as it is thinking about relative to when we're going to be planting the next wheat crop, right? I mean, this is sort of Kelsey's here, but thinking about the time period that we have to have ahead of that next wheat crops in order to prevent infection. And so, yes, it may be true that we would like to wait a little bit longer to allow a greater flush to come up. But at that point, you may be actually too late to do much good for next year's wheat crop. And so thinking about getting that control done ahead of the wheat crop. Would you agree with that, Kelsey?
Yeah, that's what we recommend. So those mites, they have a really short lifespan. They only survive about 10 days. They go through their whole life and that's it. So really, if you can get that volunteer controlled really within 2 weeks to make sure you're safe of planting, we know that we can kill off that mite population, right? You want to be 2 weeks and you want to have volunteer controlled within a pretty good distance of where you're planting your fall crop.
Maybe not so much for volunteer wheat, but for the diseases that come with volunteer wheat, finding varieties that are resistant.
I call that more of a management tool. And I think whenever we're talking about volunteer wheat and the wheat streak mosaic virus, the reason it's such a hard pest for us to get a hold of is it does not depend only on us. If we are doing a good job controlling our volunteer, and again, we want those plants to be dry and dead at least 2 weeks before our new crop is coming out. But we have a few tools that do not only depend on us. We can be doing a good job of controlling volunteer in our own fields. If our neighbor is not, there's a very high chance that volunteer might be just being held up there. So what are other tools that we may have to help kind of combat this disease, right? And there has been a lot of effort in breeding varieties that have some level of resistance either to the virus itself or to the vector of that virus, to the wheat curl mite. And so if we would talk a little bit about those varieties, the ones that are being bred with resistance to the virus itself, we call that resistance WSM2. So it's a gene that confers resistance against the virus itself, meaning that perhaps the plant will get infected, but not demonstrate to a lower intensity some of those symptoms, right? Some of those varieties, K-State has a KS Dallas, Oakley, and Joe that are KS Hamilton as well, that have that WSM2 gene, and that can help as a tool in that toolbox. Guardian is a variety from Colorado State University that also has the WSM2 gene. Now, this resistance is interesting for us to think of it as it is temperature sensitive. For most of these varieties that I mentioned, when it breaks 65 Fahrenheit threshold, so hotter than that, the resistance will be broken down, meaning that the resistance is not there anymore. So we have a window there in the winter where in the fall, when it's less than 65, and in the spring, before it gets more than 65 again, where we're quite well protected with those genes. Now, a couple of these varieties here, KS Dallas and KS Hamilton specifically, they can go up to 70. So we're expanding the window of protection a little bit. In 5 degrees on each side can give us a few days more on each side in terms of protection there. I also mentioned that there is the resistance against the wheat curl mite itself, in that the wheat curl mite will spread slower in some varieties that are less preferred. And many of these varieties, they actually go back to a background of TAM 112. So many Colorado varieties actually, for example, Bird or Avery or Langin, and they carry that resistance against the wheat curl mite itself. KS Western Star is a K-State variety that has that resistance as well. Whistler, Canvas, Guardian, Crescent AX, Incline AX, and some other varieties, mostly, again, coming from that Colorado program with that TAM 112 background, that actually they will slow down the spread of the wheat curl mite. Guardian is the only one that combines WSM2 gene plus the non-preference by the wheat curl mite. There are also a few varieties that they don't have anything that we know of in terms of having a resistance gene, either against the mite or against the virus itself. But for some reason, they tend to handle those conditions better than others. We don't say anything handle them very well, but they handle better than neighboring varieties. And some of those varieties that we observed in many of our trials that were infected by wheat streak mosaic, they were, for, again, some reason that we don't know of, handling those wheat streak mosaic infections better, including Rockstar, SY Wolverine, LCS Photon AX, LCS Helix AX, and LCS Julep. So that's kind of like a short list there. But again, I would call this a tool to manage that resistance, not necessarily something that will be bulletproof.
That was K-State's Romulo Lollato, Sarah Lancaster, and Kelsey Andersen Onofre. And Kelsey continues this conversation after the short break. You're tuned back into Agriculture Today, and we continue our conversation on volunteer wheat with K-State wheat pathologist Kelsey Andersen Onofre, as she reminds growers why it's important to keep in mind that there are many different viruses.
When we're thinking about the viruses and resistance, it's really important to remember that we have multiple viruses out there. So something like Guardian, which does have WSM2, it does have that CMC4 curl mite gene. So it does, theoretically have really good protection, at least up to a certain temperature. It is still susceptible to Triticum mosaic virus. And we've seen examples of that recently. So, there are a couple of virus varieties like a new K-State variety, KS Territory, that we know has WSM2 and is a bit more tolerant to Triticum mosaic virus. But that's just something to keep in mind that again, none of these are perfect. And we do have multiple viruses out there, which does complicate the issue.
Kelsey, talking about multiple viruses, if producers want to get their field tested, where can they do that?
So K-State has a plant disease diagnostic clinic that we have here in Manhattan, and we test for all of these viruses. So we do test for wheat streak mosaic, we test for Triticum mosaic virus, and also high plains disease, and then a couple of the other wheat viruses like barley yellow dwarf and soil-borne mosaic virus. That's one we sometimes see early in the spring. So this fall timing, if you do see those classic symptoms of wheat streak mosaic or early in the fall, it is good to get a test at that point. That can kind of indicate how severe the yield loss could be that you expect. So if we do have fall infections, typically we do expect larger yield losses. So, you can always send us a sample and get that tested proactively, even through the fall and especially in the spring as we see those symptoms pop up.
Kelsey, we mentioned the lab at K-State, but there's also your extension offices are able to help with this as well?
Yeah, absolutely. So your extension office and your local extension agent, K-State extension agent, they're great resources. If you suspect you have issues with wheat streak mosaic, you can bring samples to them, and they can also provide weed management and variety selection information too. So make sure to reach out to your local K-State extension agent.
That was K-State wheat pathologist Kelsey Andersen Onofre. I will link the resources that they mentioned in today's show notes on agtoday.net. We're now going to be joined by Associate Professor in the Department of Clinical Sciences in K-State's College of Veterinary Medicine, Brian Lubbers, as he discusses things producers need to keep in mind if they walk out and see dead livestock in their pens. Brian, thanks for joining us today.
All right, thanks for having me.
You walk out into your pen and see deceased animals. However, before we even get to that point, it's important to put together a plan.
The plan's actually the important part of it, right? And what we usually find is, that scenario happens where someone walks out, they have a dead animal, and there is no plan, right? And there's actually nobody ever wants to see that, right? And so, but there are some things I think we can get out of that situation that having the plan will help us do a better job. And so, really, the first thing is that animal provides us the opportunity to get some information. So what was the cause of that mortality? Was it an infectious disease where now I'm worried about other members of my herd? Is it kind of a one-off thing? What exactly happened? And so the first part of that plan is how to kind of do the necropsy. And I encourage people, we often think that, we have to call the veterinarian out to come and do a necropsy. And yes, that can happen, but it's also possible to train producers to do their own necropsies too. And really what we're trying to do with those is catch more information as the veterinarian's not available, especially in the summertime. If you can't get a veterinarian out very quickly, decomposition happens and so it becomes, if you can't get somebody out in a matter of hours or so, it may not be worth it to try to do much with. And what I always encourage people to do is take lots of pictures, right? And so take pictures before you move the animal so the veterinarian's able to kind of see what environment it died in. And then we can teach a very simple, basic necropsy that people can do in 20 minutes or so, plus or minus, and take pictures as you go along, right? And depending on what you see as you kind of do that necropsy, there may be some cases where a veterinarian would say, this might be one where we want to take some samples. If we suspect it's a toxicity, again, if we suspect there's some infectious diseases going on, there might be different types of samples to collect to kind of help us narrow down. And really, I make a big deal about the necropsy because typically what happens is if we don't have the plan, we don't do the necropsy, we lose all that information, and then lo and behold, another one happens a couple weeks later. And the question always is, are they related? Are those two animal mortalities related? And if we've already started that process, we're gathering information from day one, we're just that much further ahead of the curve.
A little bit of difference there if you walk out and see one animal dead versus maybe 10 or 15.
Yeah, that's a much different plan, right? And the level of criticalness kind of goes up, right, when we have those mass events. And again, we can still see the same causes. It could be, we see individual animals that die from lightning strike, and we see group mortality events, right? We see the same with heat. We see the same with infectious diseases. We can see the same with toxicity. So the number may help us think about certain causes as more or less likely, but even then, that information is still just as important. Now, as far as doing a necropsy on a larger number of animals, probably not necessary. If it's all seems to be related, we can probably pick one or two, maybe three representative animals to do the testing and sampling on. But then we get into, the difference at that point becomes some of our disposal options, right? And so if it's one animal, we kind of go maybe down one path as far as disposal. But if we have these mass events where animals have died off, then there's different set of regulations that come into play. The recommended disposal methods are a little bit different. And in Kansas, we fall under regulations by the Kansas Department of Health and Environment. And their order of preference is to render first. So that's the first option, depending on how many animals you have. And then composting, landfill disposal, on-site burial, and then incineration is kind of the last option. And there's pluses and minuses to all of those. Cost, availability of rendering may not even be an option if it's a really large number of animals. The impacts on the environment, all those things you just kind of balance all those out. But really at that point, you'd be working with them to figure out what would be the best option.
But a lot of information there to maybe have thought through before you hopefully don't get to that point, but in case you do.
Yeah. Nobody wants to get to that point, but certainly having that in place and having a plan When you deal with the Kansas Department of Health and Environment, it may require permits, which you may be able, or at least have a plan in place with them ahead of time so that we're not kind of scrambling last minute, especially on those larger mortality events. But even the one dead animal scenario, it's nice to have that in place so you know exactly what you're going to want to do. And certainly we want to, whatever our method of disposal, we want to get our necropsy done, and whatever our method of disposal is, we want to get that done as quickly as possible just to kind of avoid, smells and things like that.
And we talk about cattle a lot on this on Ag Today, but it's really for every species related to agriculture.
Yeah, sure. I mean, all of those principles apply, whether we're talking about necropsy or disposal, they apply whether we're talking about cattle, sheep, goats, all of that would apply.
And one more you wouldn't walk out and see just a euthanized animal, or hopefully you wouldn't. However, the disposal for that type of death is different.
Right. Yeah. So we'd think about that differently. And really what we're talking about is when rendering is an option, right? And so we want to make sure that we're not, if we have an animal that's been euthanized with a barbiturate or whatever, the veterinarian's done that, it's an anesthetic overdose, that can hang around in the tissues. And we've actually had some cases where pet food became contaminated from that euthanasia agent still present in those tissues. So if rendering is an option, then we might, again, part of our plan should be not just when you find them, but if you have an animal that doesn't have a chance of recovering, we want a humane euthanasia. And so how are we going to do that? And keeping in mind, yes, what our disposal option at the end will be.
Talking also withdrawal time is really important to the ag industry. However, not as big of a deal when worrying about disposal of passed away animals.
Yeah, so the withdrawal times apply when that animal is entering the human food supply. And so we're not as concerned, if we're talking about antimicrobials, biologics, for a lot of these like composting, burial, things like that. Almost no concern for rendering. Depends a little bit on the product, but for the most part, it's not really a major concern for us because obviously those products don't go into the human food chain.
So for producers who maybe don't have a game plan put together yet, what's the first step you'd recommend they do?
Always first step, talk to your veterinarian, right? Because we can sit here and talk about generalities about a plan and stuff, but all of these things might be slightly different depending on your situation, your geographical location. So they're the ones that can really individualize that plan. Also, with the necropsy, some veterinarians are much more comfortable with training and evaluating necropsy from photos. Others may want a more hands-on approach. So it may be the difference. The first option is have the vet do it, and the second option is producer do it. In other situations, it might flip-flop, and they're more than happy to evaluate those.
Brian, I appreciate you coming in today and sharing with us some information about a topic important to producers.
Yep, thanks for having me again.
That was Associate Professor in the Department of Clinical Sciences at Kansas State University's College of Veterinary Medicine, Brian Lubbers. I will link the resources that he mentioned in today's show notes, which you can find on agtoday.net. We're cutting to a short break now on Agriculture Today, but when we come back, be joined for our weekly horticulture segment with Raymond Cloyd.
This is Agriculture Today. Along with Shelby Varner, I'm Jeff Wickman. K-State horticultural entomologist Raymond Cloyd says squash bugs, blister beetles, cucumber beetles, and squash vine borer are all currently active in Kansas. Raymond, maybe an update on bagworms to begin with. Finally reached that point where it's really too late to spray?
Yeah, Jeff, at this point where the bagworm male and females are going to cap the top, we call that the green or brown cap period. Unless they're coming out of the bags and feeding, any sprays are going to be very ineffective or result in low mortality. So at this point, it's probably best to go in and just hand remove the bags because at this point, spray applications are going to be less effective than they were early on when they were, like we always recommend, an 1/8 to 1/4 inch long, and the caterpillars are coming out feeding quite regularly. So at this point, that's the strategy for dealing with bagworms.
In terms of squash bugs, what are we seeing there?
Squash bugs are out. We're seeing the gray fifth end stars, the adults. A little bit on squash bugs. The overwinters adults, they come out, they mate, then they lay their eggs, the red, golden, or dark red eggs and leaf underside. The nymphs will emerge, you have 5 end stars, and then they become adults. And, of course, everything is in leaf underside. So if you're thinking of spraying, or spraying is one of the recommendations, is you have to get in leaf underside. So you can squish the eggs or apply a horticultural oil, a mineral-based oil, to kill the eggs and the early end stars, the nymphs, that is, that are out there. Once they become adults, they're very difficult to manage with insecticides because of the very waxy cuticle the adults have, which is very similar to grasshoppers. There's always the hand picking, the vacuuming, other means, and there's an extension publication you can get online that details some of these management strategies or plant protection strategies to deal with squash bugs. Remember, they do vector diseases and they vector a virus, and that can result in death of the squash plants.
Another thing we're seeing are blister beetles.
Yes, we're seeing a wide variety of blister beetles, but the main one is the ash gray blister beetles feeding on eggplant, causing substantial damage. You have to be careful with blister beetles because they will emit a material called cantharidin if you touch them. It's sort of a defensive compound, but they feed an eggplant. They can basically defoliate a plant given enough time and the numbers are there. If you want to go out and hand pick them, you can wear rubber gloves or leather gloves, pick them off, put them in soapy water, and kill them. There are sprays that are out there, but you're going to have to make multiple applications to kill the adults during this time of year.
Also, cucumber beetles, another thing that we're seeing in the garden.
Exactly. Another vegetable pest, the cucumber beetle. We have the striped cucumber beetle and the spotted cucumber beetle. The spotted cucumber beetle right now is feeding on the flowers of cucurbits because it's attracted to the volatiles that are given off, and that could impact the pollination. So you have to be cognizant of that, and you spray with some of the common materials. If you have any questions regarding that, you can contact your extension office or Department of Entomology at Kansas State on what materials to use. You have to be careful because you don't want to impact the bees. So you have to spray early in the morning or late evening to minimize any direct or indirect impact on bees, especially honeybees or bumblebees.
And finally, squash vine borer.
Squash vine borer, if you go out to your squash plants and you notice 1 out of 4 is wilting and you've given them water, you might want to check the base. If you see a green goo, sort of like a split pea soup type of material at the base of the plant, you might have squash vine borer. And a squash vine borer is a larva that tunnels into the squash plants and moves upward and cuts off the plant's ability to get water and nutrients, and that's why you see that wilting. Once they're inside the plant, it's really too late. Okay, so you have to think like next year, what we recommend is to watch your squash plants for red eggs. The female, which is a moth, lays red eggs at the base of a squash plant. You can squish them, apply a mineral oil, but when those caterpillars or the larvae emerge and tunnel in, it's too late. Another recommendation is after the end of the season, remove all debris. What they'll do is after tunneling in the plant, they'll go down to the base and they'll enter the soil to overwinter. So sanitation, removing all the residues and debris, and this is the same for squash bug, will alleviate or remove any larvae that are still in the plant, and for squash bug, remove any overwintering sites.
That's K-State horticultural entomologist Raymond Cloyd with an update on some of the active insect pests in Kansas. Again, for more information, contact your local extension office or K-State Entomology. And that'll do it for this edition of Agriculture Today. For Shelby Varner, I'm Jeff Wickman. This is the K-State Radio Network.